Today we're going to hear from the VertNet team, and so David Bloom is going to introduce the team and then the project, so I'll just turn it over to you, David, and you can tell us all about VertNet and all of the wonderful ways it's going to help us discover all the specimens that we care about.
Thank you. It's probably good you didn't use that 2-page bio that I sent to you. So first of all, I'm not Carla Cicero. She sends her regrets and would certainly much prefer to be here than where she is right now. But she had a family emergency that she needed to take care of. As a result, I will do my best impression of Carla. And I will also do an impression of a VertNet intern who's based at the University of Colorado later in the presentation. Carol Spencer will also be giving an impression of Carla a little bit later on. There will be a lot of tagging and a lot of teaming going on up here, but feel free to jump in with questions anytime. My name is Dave Bloom. I'm the VertNet coordinator. Many of you, I suspect, are familiar with VertNet, as it's been based here at the MVZ for the last 5 or so years. A couple of people in the room I just want to call out who are on the core team that are around and that you will see on a regular basis or semi-regular basis. Michelle Koo is over here, Carol Spencer, who you'll see in a few minutes, Byrne, who you'll also see in a few minutes. Aaron Steele is back there. He's our lead information architect. Is Robin in here too? Robin Kraft back there, who is working with us for a period, and then even after we're done paying him, he will probably continue to come in and work in the GML just because he likes the chairs. And I think there are certainly a number of you that have helped with the project in varying capacities over the years. But let me just go ahead and get started, give you a sense of, there we go, what VertNet is. VertNet was created as a result of a large-scale collaboration in 2008 to address pressing needs of the 4 vertebrate networks that have existed since 1999. Not all of them, but beginning in 1999. How many of you are familiar with either VertNet or one of these 4 vertebrate-based networks? Okay, how many of you actually used one? That's pretty good. More of you are familiar than used, but that's okay. There is a long history with these networks in the MVZ, particularly with Manus, HerpNet, and Ornis, and that much of the creation of those networks was based here, as well as some of the impetus socially to get these networks going. And all 4 of these, as well as VertNet now, is really it's a cumulative effort. The idea is both to solve technical as well as social issues that arise with sharing data, so that the data can be free and accessible and discoverable. What VertNet is about is combining these 4 networks into a single network or a single portal where all of this data can be searched at one time, as opposed to having to go to multiple portals to get various types of vertebrate data. I should add that all the data in these networks goes to GBIF, which is great. All of the data that goes into VertNet also goes to GBIF. That means that if you are looking for things on bison, which is the North American node of GBIF, all the VertNet data is there as well. We'll talk a little bit more about portals later on, but and what you might find in various portals. But that's the nitty-gritty for our history. A couple of things about VertNet. We're now into our 3rd year as an actual, real-life, honest-to-God thing. With NSF funding, we're in the 3rd year of a large grant to build VertNet out. Right now, we're looking at about 409 collections from 167 institutions globally. Just to give you a sense of how we get used, we served over 2 billion records last year. I think that was more than McDonald's. I was just so wanted to say I'm more than it, but just kidding. At any rate, right now the VertNet portal is in beta. We'll talk about that. We'll explore that in a little bit. But when we're up and running, we should be at about 150 million records from those 167 institutions. So it's getting big. which is great. And that has the potential to be growing depending upon the affiliations within VertNet. It could be growing at a rate of about 40 million records annually once this thing gets going, which is pretty big. The other piece that I would be remiss to mention is all of the training that we do. So we're building a data network for sure, and we're making data available, but we're also training people all over the world, both to clean their data, to georeference. Many of you were probably trained by somebody in this room at some point in time to do georeferencing in particular, but biodiversity informatics trainings at the University of Colorado, a whole variety of opportunities there, upwards of about 1,000 people globally that we've reached out to and helped to become contributors to their local communities. Where does the data come from? Well, it comes from all over the place. Mostly we just take the stuff that Beth and Josh put in the room on the other side of the MVZ. They're quite busy, especially if they're going to hit 150 million records. But we get this stuff from all over the place. It's large museums, it's small museums, it's collections with standards, it's collections with no standards. If they're ready to publish, basically if they're ready to digitize, if it's been digitized and it's in a format that we can handle, it can go into VertNet. It's not, of course, quite that simple, but it is nearly so. We've done a lot of work over the last couple of years to simplify the process for participation and break down barriers so that anybody who's got a collection and data and is ready to share it can participate, which is an important piece to what we do. Why do we have VertNet? Well, one of the big issues is growth. In 1999, FishNet started down here with 6 institutions, and they had their issues. They've had their issues, I should say, over the last couple of years. Manus followed, based here at the MVZ in 2001 with 17 additional institutions. And over time, with the additions of HerpNet and Ornis, it's just grown to the point where the 2.5 of us that deal with these institutions to move their data into the portal simply just can't keep up. Right now we're dealing with 44 institutions that are up and online and ready to go using our publishing methodology, which is called the IPT, or the Integrated Publishing Toolkit, that was designed with our help by the Global Biodiversity Information Facility, or GBIF. But you know, we're at 409 collections right now that are either up or waiting to get up. I think we still have something like 270 collections that we need to mobilize that are just waiting. Some of these, many of them are already in one of the other vertebrate networks, but because those vertebrate networks are basically sunsetting and they won't be supported at the end of this calendar year, everything is going into VertNet. So over time, this will be your one-stop for vertebrate data. So growth is a big deal, but growth is one of the biggest problems. The old networks, which you can see down here, this attack of the killer post-its, every line, every square, everything in this right here is a potential point of failure. It's because it's an old distributed network, and so it goes from the request to a server, to another server, perhaps even to another server, and then back, one institution at a time. That's how the old digger-based networks used to work. HerpNet and Ornis are now in their second phase, so you'll see if you use them, HerpNet 2 or Ornis 2. FishNet 2 is also using a new protocol, and that makes it much faster, but it's still not exactly where we need it to be. And so with all of these potential places where things can fall down, we needed to change how these networks worked so that we could be more performant, we could be faster, we could aggregate data, the whole variety of issues and challenges that confront this type of architecture that Aaron and Robin and others are dealing with right now. This is only part of the new network, but basically it's pretty simple. You get an original data source, they send us a text file with their stuff, they put it in a Dropbox. We then run it through a migrator that I'll tell you about. We send back a quality report. They either fix it or they don't. And then when it's ready, when the collection says it's ready, we publish it to our IPT. And then that is then harvested by a whole set of software called Gulo, which then runs all that stuff through and makes it available in the portal, makes it available via visualizations and so forth. And so it's much simpler. It's also way faster. There isn't any query that you can put in right now and then go to lunch and come back and see that it's not working any longer. That doesn't happen, which is great. It's still breakable, but it's still in beta, so we want you to break it, by the way. But it's much faster, and we've also solved a lot of problems because each of these lines, each of these squares represents something that needs to be maintained, and that costs us money, that costs the institutions money, and without all of these squares, you basically just need to have your database and be able to attach or connect to the internet to send us something to Dropbox, and you can be in. There are no more servers to maintain, no more IT hassles, at least for the most part. And so it's basically free for participation now as opposed to, yes.
Does VertNet continually query all these databases and get new material, or you are dependent on them to give it to you?
So, yes. So what happens is that...
Yes, which?
So what happens is that we set up a separate IPT, which has their, we'll look at these a little bit later on so you can see where they are and we can prove that they exist. It's basically a spreadsheet, CSV with all of their data in it that they've approved for publication. If they want to update it, they can send us a new spreadsheet, a new text file. We'll run it through the migrators, clean it up as necessary, and republish it. We then, on the other side of that, as a separate process, can harvest this on a regular basis. I think we've been talking about doing it once a week or at a minimum once a month, so that anything that's updated by the institutions happens separately, and then the harvester itself just goes to the publication CSV as opposed to having to go to 13 servers. So it never goes back to the source, it just goes to that IPT that we've set up and harvests from that. So if that's updated, yes, that gets updated every time we harvest. There were a couple of challenges that I mentioned. We had technological challenges to deal with as well as social challenges. Human biodiversity is a really big issue for biodiversity networks because there are lots of people that understand things and lots of people who haven't yet understood things and lots of people who don't want to understand these things and how they work. And so some of these people are collection managers, some are curators, some are students, some are just people out in the field. But what we've been trying to do is work with all of these people to meet them where they are and try to move their data and or their experience in finding data forward. Technologically, I mentioned there were a number of issues in terms of speed, scalability, performance, and so forth. But probably the biggest issue for us technologically is data discovery. How do you find the data? Even if it's in a portal, how do you find it? Because you can use a few keywords right now, maybe click a couple of boxes here and there, but are you really finding everything that's in that portal? That's a good question. Well, one of the ways that we're dealing with this is that we are using something called the Darwin Core, which is, the Darwin Core standard, which is becoming very quickly the global standard for sharing biodiversity data. There are 159 fields or headings for data that tell you the who, what, where, when, and about of a particular collection event. That can be a physical specimen and it can be an observation. So it also involves tissues and other sorts of things that we can get into extensions, but basically we have a bunch of opportunity here to organize a bunch of data in the same way. So everything that goes into the various vertebrate networks, as well as into VertNet, as well as into GBIF, now into iDigBio. If you're not familiar with them, that's the new US hub for digitization. They're all using Darwin Core so that when you get data out of a portal, it's all in the same format, regardless of the source. This is important, as we'll see in a little bit. Some of this may look very familiar to Rory and to Michelle. Carla was looking through a whole bunch of information for a grant that they were working on, a project that they're doing, to just kind of get a sense for what the data is like. And for scrub jays, there are 204,000 plus records in ORNIS and VertNet on this particular species. But, unfortunately, or fortunately, depending upon what makes you happy, there are 37 unique scientific names for this one critter. 113 unique preparation types. 8 unique countries, including Mexico with a question mark. A lot of locality unknown. That might be the 197th country. 60 unique states. 4 variations. And the point being is that what she discovered, what they discovered, is that there's a lot of variation in the data. This is good. and it's not so good. Dealing with data inaccuracy is something that we can deal with. If Missouri is listed as a country, that's something that we can fix relatively easily. Also data incompleteness. If we have a county and some lat longs, we can usually figure out what state it's in pretty easily. If we have a state, we can tell you what country it's in. If we have a province, we can usually do the same thing. Data inconsistency, however, has proven to become a much, much bigger issue than we could have imagined. So we developed these migrators, software packages that we can take data in when people send it to us and put it in our Dropbox. We can take that, customize the migrator, and help them to clean up their data. And what these migrators do is they identify empty cells or empty fields that can be filled in. They tell you when something should be a verbatim cell as opposed to like a decimal lat long as opposed to a verbatim lat long. We can deal with some taxonomic issues, particularly higher taxonomy, higher geography, a whole variety of issues that we can then identify very quickly, send back to the original source. They can fix their data, and many of them do fix their data, and then prepare it for publication. So we can clean up quite a bit. Where there are standardized vocabularies, that's really super easy. Where there aren't, this is going to be years and years of work for those of you who are at the beginnings of your careers. And for those of you who are not at the beginnings of your careers, you may relish the thought that you have left something for people at the beginnings of their careers to do. By way of example, how many countries are there in the world?
176.
196. There are 236 recognized by the ISO because of abbreviations or name changes, a la Myanmar, Burma. Sadly, even after cleaning up 2.7 million records, but we still found 4,900 different countries in this very small subset of data. Things like Lower Amazon, that's a good one, or Africa by inference. These are people in many cases with PhDs that are filling out these forms, so just be wary. We can, of course, talk about sex because this is a university setting. So how many ways are there to denote the term male as far as gender equals male? How many ways? 35? That's the best answer I've heard in giving this presentation or parts of this presentation for 2 months. Who else? How about female?
3.
How about 189 different ways to express male, 184 for female, and 331 that are unresolved. This is a field that has standardized terminology. We shouldn't have this kind of issue. Hard to know exactly how to deal with that. Preparations, well, I won't even get into that, but there were, we went through these 2.7 million records. We discovered almost 11,000 distinct preparations. Of those, there's some sort of combination of almost 3,000 distinct terms. And from that, Carla, bless her heart, went through and discovered that there were probably 26 actual terms that one could use to describe tissue with some accuracy. This is all by hand, there's no standardization. It was, of course, very interesting to note that one could find vodka in here, and you can search for that in VertNet. Surf, STST01, not exactly sure what that preparation is, but the point being that there is still a lot of work for us to do as we move forward. So what I want to do now is, we have a couple of other things to do, but I want to introduce one real person and one alter ego. One of the things that we did this summer through NSF, with NSF funding, is we brought on 2 interns. One was based here, the other was based at the University of Colorado. And they were charged with working with us through the summer to try to make a whole variety of interesting things happen. I will pretend to be a Victoria Tersini in a few minutes, but I want to introduce Byrne, who has done some really cool stuff, particularly for those people who are into amphibia here, dealing with Chytrid. And so she's going to give a little presentation about what she did over her summer vacation. And then we'll come back and we'll look at the VertNet portal and do a little bit of demonstrations. So, Allie, by the way, is a recent graduate of Drexel University. She is a transplant to the appropriate coast and is now currently in the process of seeking out graduate opportunities hither and yon. So, Allie, go for it. Yep. I'll set you up. Thank you.
Hello everybody, my name is Byrne, and yes, I'm the VertNet intern in the flesh. So this summer I had a really awesome position in which I got to create my own research project based on my interests that also contributed in some way to VertNet. And I ended up doing two projects actually, one which I'll present today and the other which we don't have time to get into, but I'll mention. I wrote a script in R that flags outliers based on a species range shapefile. And so that script in R is on my GitHub, and if you're interested, come talk to me about it. I'd love to chat. But today, let's get into my project titled Hitching a Ride, Did Herpetologists Introduce Chytrid to Southern Mexico? Start with a little background information, because we're not all herpetologists here. Chytrid is short for Trachochytrium dendrobatides, and it's a pathogenic fungus that infects the skin of amphibians, and it causes a disease called chytridiomycosis, which is very deadly, and it's been causing rapid amphibian declines around the world. But it is interesting to note that Chytrid doesn't kill all amphibians. Some can carry this fungus and not be affected by it at all, and some die quite rapidly. This map shows current distribution of BD. And a dead frog. Alright, so one big mystery about Chytrid is, where did it come from? The most well-supported hypothesis on the origin is that Chytrid was introduced to areas in which it was new from areas in which it was endemic. And along this same line of thinking, a lot of people have gotten behind this idea of the out-of-Africa hypothesis. So in the 1940s and 50s, Xenopus laevis, the African claw frog, was used as a pregnancy test. They would inject urine into the frog, and if it started to lay eggs, then the woman was pregnant. So there was a high demand for this frog, and it was widely exported out of South Africa, where we know that it was carrying Chytrid. Xenopus is one of those carrier species, by the way, that doesn't really die from it. So this leads me to introduce my hypothesis, which is that Chytrid fungus was introduced to southern Mexico by hitching a ride on the field equipment of a herpetologist. So, important for my project, physiological limits of Chytrid. Now, we have yet to discover, or there doesn't exist, a resting resistant form of Chytrid that would be able to travel long distance. So the capabilities for it to travel are limited by the zoosporangia and the zoospores and what it requires. So we know the optimal temperature for Chytrid is about 17 to 25 degrees Celsius. Optimal pH is 6 to 7. And so this graph shows temperature. This group here shows that range, 17 to 25. The middle line is 10 Celsius, and then this line here is 28 Celsius. So there's a pretty fine line that once you cross it, Chytrid dies pretty rapidly. Chytrid survival outside of its host. In the laboratory, we found that Chytrid can survive for 3 months in moist, sterile river sand, which is important for my project. Chytrid can also survive in bird feathers, as well as for 7 weeks in water. So why are we talking about southern Mexico? So this area is of interest because there's been a lot of research done on this wave-like spread of Chytrid through Central America, and it's all started up here. This figure is from a paper published by Tina Chang in 2011, and she was swabbing specimens right here in the MBZ and found the earliest known Chytrid, or positive Chytrid specimens from 1972 and 1974. A paper published this year by Phillips and Puchendorf took that one step further, and from the date of decline for some of these species and from those dates I mentioned earlier, was able to create 95% confidence intervals for when Chytrid was possibly introduced to these areas. And so this paper was really important to me. This is the time range that I'm looking for, so 1959 to 1974. looking at some genetic data, this recent paper took that spread from Mexico to Panama and sort of extended it further north, and found that as you go from California south to Panama, the genotype of the BD decreases in heterozygosity. And so that supports this idea that Chytrid was subsequently introduced and then proliferated as you go from north to south. This here is a neighbor joining genotype tree, and yellow, it's hard to read, yellow is Mexico, and it's closely related here to Panama, California, and Arizona genotypes. So now we'll get into what I did for my project. I started out in VertNet 2 because 2 months ago when I was doing this is the best that we had. There's VertNet 2 hosts 51 institutions currently, and so I used a geographic search, used my date range up here, but in the very near future we're going to be able to use VertNet for all of this. So same functionality, we got a geographic search, years, put in herp, and very soon we'll be getting more than 51 institutions worth of data. So from that I was able to get some collector names. I parsed out those names, and then I went back to the portal, plugged them in, and I ended up getting a set of data for each name. But I don't really want all that data. I just want the collectors who went somewhere of interest within 3 months of going to Mexico. Because remember that 3-month survival of Chytrid. And so from that I got 55 collectors, 347 events. Alright, that's the starting point. So then I encountered some problems with the data that were mentioned earlier. Here we have DR Paulson, same day, 4 different countries, so unlikely. So we have to kind of dig a little deeper to find out which is the correct date here. I've been using catalog cards in the museum, field notes, looking at the publications to try to figure out which is correct. And when in doubt, I had to exclude the record. Here's a look at a catalog card. For this record, a little example, here it is, and we look at the date, date's 66, 67 here. Oops, had to delete that point. Similarly, I used the field notes. Here's a little excerpt from J.F. Lynch's field notes, who, by the way, kept excellent notes. So he mentions he left Berkeley on his way down to Mexico, and then he stopped in the Mojave Desert, where it got over 100 degrees, even at 2100 hours. So that is not exactly an ideal situation for Chytrid. So I was able to eliminate this event from my list. So from this giant list, I had to try to pull out the most important ones, the most likely events. And I ended up developing a ranking system. So I kept it simple, 3210, the top 25% got a 3, bottom 25 got a 0. Here are the categories I used. First, how many days were between their travel, and how many locations they stopped at. That's simple, got it straight from the data. Next, I designed a Chytrid suitability score for the origin. So I took into account the mean temperature of the summer months, the pH of the soil, and the soil drainage. That was kind of an onerous process. I used the Harmonized World Soil Database, and then I had to reclass it in ArcGIS, and then combine all of my rasters, and then I extracted a value from that raster in R to get my value that I then compared and scored. For number 4, I looked at how close they landed to these 3 sites that we talked about earlier. So just a simple multiple ring buffer in ARC, and then I converted that to a raster, did another extract. Next, I wanted to look at 2 known carrier species for Chytrid. One is Radicata biana, the American bullfrog, and Xenopus that we talked about earlier. So the way I did this is I pulled data from GBIF pre-1974, looked at the density, or calculated the kernel density in ArcGIS, created a raster, same kind of extract process. And lastly, I looked at the proximity to known positive sites. So this is where the data is really limited as far as literature goes. There's a lot of swabbing going on, but not a lot has been published, so kind of limited to a few points. But yeah, same extract. And then from that, we have a max score of 21 points. Here are all the points that came up possible, so 347. And I went through and started eliminating them. So first I removed points from areas where we know Chytrid is absent, such as West Africa here. A recent paper found that Chytrid doesn't live over here, surprisingly. Next, I eliminated points that are in unsuitable conditions for pH and temperature. This might not be for them to live there, but for them to be traveling in the soil, it would be unfavorable. So, clipped out those points. If you remember from those confidence intervals, they were a little different for each site. So then I sort of clipped around each. I used whichever site they were closest to, and then I clipped out the points that landed outside that interval. And finally, I clipped out points that landed outside this buffered area altogether. And I ended up with 126 points. All right, so we're getting somewhere. Then I looked at the distribution of the score, and I compared that to how many days between. I picked this out as the most limiting factor here because for every day that passes, Chytrid is more likely to get too hot, to dry out, to basically die in transit. So my most interesting points here are on the low end of days, high end of score. These are my top 15 events. So let's look at this on the map. So we see a high concentration here, and that's probably from the Rana, Catspiana, and Xenopus, and also the right temperature, pH, all that stuff. It's all coming together down here in Southern California. So we're going to take a closer look at some of these events. This is my number one event. Score is 17. It's from 63. They go from San Diego to Guerrero in 10 days. We'll look at what they're collecting in San Diego. A night snake. Okay, well that's interesting. Not really a Chytrid carrier. And then, thanks to Google Street View, we can actually see what it looked like where they were collecting. All right. So yeah, not the best environment for Chytrid. We'll call this point unlikely. Next, we look at our number 2 point. Score is 16, going from Los Angeles to Oaxaca, 18 days, 69. All right. Catching amphibians this time. So, all right, makes it through that test. And then let's look at the map. All right, so landing, it's not right in the middle, but I think we have to call this point possible. All right, so last but not least, let's look at our number 3 point. Score of 16 in 1972. San Diego to Veracruz, 19 days. Catching salamanders, all right, that's interesting. Now we're going to walk through this whole sort of trip that this collector took. Started up here in 72, 19 days later landed here, 21, 22, 23. And when we get to 24, it's interesting because this is the specimen from that Cheng paper. It's the earliest BD positive specimen, and it was collected on this trip. So we kind of have to think about what that means. Does it mean it was unlikely because Chytrid was probably there when they got there, or does it mean that maybe their equipment was contaminated in some way and that's why it's coming up positive? I think the latter seems unlikely because the Cheng paper actually swabbed a bunch of points from this day, from a lot of these days beforehand, and all of them were negative. So I'm thinking this one is an unlikely, but we have to look further, so we gotta call it possible. So, in conclusion, only 2 out of my top 15 events were deemed possible. And they both definitely require further investigation, which I look forward to getting into. And I was able to highlight some prime areas where BD could have come from, mainly California. And in contributing to VertNet, I definitely demonstrated the importance of having access to millions of specimen records. Without VertNet or its predecessors, I wouldn't even be able to think about doing this kind of analysis. Highlight the versatility of the portal. I was able to get exactly what I wanted, when I wanted it, and surface some of the data quality issues we talked about earlier. And more directly, I georeferenced a ton of records, so that'll go right back into the portal. In the future, I definitely want to refine my ranking system. Giving equal weight to all those different factors, probably not the best way to go about it, but it was the simplest, the fastest. So I'm going to look into that, comparing historical swabbing results to the data set. There's a lot of swabbing going on, but that data is not able to be accessed right now. Wait till it's published. Looking at the strain of BD found in these historical specimens is definitely important, so we can compare that to where it might have come from. increase the size of the data set. So as VertNet grows, more collections want to be a part of it, so more data becomes available. And then going through and eliminating some of these points in the ways that I outlined earlier. And then even using VertNet to maybe look at other means of Chytrid dispersal, possibly bird, migration birds, things like that. So that's it. Thank you very much. I think we're going to do questions at the very end.
So for my second impersonation, I will be Victoria. I'm hardly going to do her work justice. She is way smarter than me. And she worked on 3 projects that I just want to bring to your attention so that if you are interested in picking up any information, you contact me or Rob Goralnik at Colorado, who was supervising Victoria while she was there. She did 3 things. One was that she worked with one of Rob's PhD students on validating taxonomy. And she and Gaurav used OpenRefine and a new tool from GBIF to call for their taxonomy. They used R in order to make that happen, and that actually works really well. That's something that has been written about both on the VertNet blog as well as on the GBIF developers blog. So both of those, you can learn some more about how that piece works. If you've got taxonomic validation to do and you'd like to automate it, this is an awesome tool. She also worked with folks at Colorado and their collaborators at the University of Florida to start clipping rasters to deal with some very much needed data. She's automating this process to improve their ability to look at layering in the Florida Plant Global Change project down there. So those rasters are available as well as her process of automating that clipping and layering. And then finally, she was looking at phenology data. She was taking the MODIS data and working with folks at NEON, which is based in Boulder also. She's created a whole workbook, an R workbook on beginning to deal with and how to look at the data for the arrival times and migration times of birds. Some really cool stuff, and as I said, stuff that I can't even begin to get into without tripping all over myself. But this information is available, so if you're interested, you can talk to anybody on the VertNet team, and we can direct you to the appropriate places where all of the scripts and packages and notebooks are. That said, we'd like to jump back to the portal, and before we conclude, give you a sort of live demo of the portal, which was released just a few weeks ago. You can also play around with this yourself at portal.vertnet.org. This is live, 44 institutions, 97 data sets. Those data sets, by the way, sometimes include multiple collections, sometimes are just one collection. Sometimes you just need to kind of check them out to see what's in there. But now it is time for Carol's impression of Carla. And so we will begin.
Okay, so a few things just to notice. So this is available for anyone to use right now. So at portal.vertnet.org, or if you go to the VertNet site, you'll see a little thing that says, please try out our portal. And again, this is still beta, so a lot of the collections that are available right now on Ornis, VertNet, Manus, and FishNet are not all on here yet. As you can see, there was only 44 publishers, and on each different you can get 51 publishers right now on VertNet and 70 or something on Ornis. So if you're actually looking to download a lot of data right now, you still probably want to go to the original portals but it's good to try this out and see what you think. So, okay, so first we're going to talk about some of the different buttons that are available, and we're going to go to the publishers. So you can see there's VertNet, searching, publishers, about, and feedback up at the top. So if you look at the publishers are the institutions or people that are making their data available. And you can see right now there's like 44 of them, so many different names. We also provide some information on what the actual abbreviation is for that portal when you're looking at the data that you've searched, how many records are available right now for each one, and you can also see how many collections are available, so that's the last number. So if we click on one of these, like MVZ, you actually go to a list of the different data sets we have available for MVZ. So most of you are familiar with most of these the egg and nest collections, the herb collection, birds, and also all the observations. Those are separate collections. And this is through all these are available through the VertNet IPT. You can see when these data were published, so referring to what Jim was asking, those will be updated when new data is put up, so we'll have a new date. And you can also see how many records, so that gives you an idea of really how much data we're serving. And if you actually go to one of these, you can actually see information. This is the actual integrated publishing toolkit. This is the metadata that goes with it. And so it gives you who the contact person is. So this was for the bird collection. So it gives Carla as the first point of contact, and then the programmers and Dave Bloom. And you can actually, up at the top, you can, if you want to download all the bird data in one fell swoop, because that's what you're interested in for MVZ, you can actually do that right from here. So you can download all the data in one, and you can do that for all the collections that are available on IPT. So that tells you a little bit about what kind of data is being published. And if you want to do a search on every single thing that was available for like the California Academy of Sciences, you could have also clicked on one of the search buttons and it would pull up all the records. Okay. So first we're actually going to do a full text search to show you what that means. So basically it searches on these words anywhere in the record. Okay. So for Vireo, and we want MVZ. And one thing you'll notice right at the top is it's actually showing you some things for the Delaware Museum of Natural History, but I'll explain why that's happening in a second. And then it shows lots of MVZ records. And it will show up this table that just says 1 of 20 to begin with. And if you want to see more, you can actually, well, you can see more, or you can hit the download button there, which is in green. That's this. Thank you. And if you want to see more information, you can also, we sort of just have a very basic amount of information that's available right here to see. So their identification, which means basically the catalog number, the taxonomy that's associated with that, the locality, which gives a higher geography to, the year it was collected. If it has a little point here for map, that means that it has a latitude longitude, so you can view it on a map. And then media is the final one, so we have one record that has media here. And that means it has some kind of associated media, like a photograph or sound or a movie. Okay, so we can go, if you click on one of these, you can actually get more information. So just the little summary gives you just the catalog number, the institution information, and the decimal lat long, and it puts it on a map for you for that one point. But if you want to find out some more information about this particular point of data, you can go to the data rights, and it actually tells you the information, the rights that this collection is given. But sort of more importantly, if you're trying to cite these data, you actually should cite data you're using in a paper according to the institution's name, not VertNet. So it tells you what the name of the institution for citation. And if you find a data problem, one will show you another way to fix data problems or to note them, but you can also write to the person who's listed as the contact. So it's a little more direct way of figuring out problems with the data and writing to the person than we had before with the old VertNet. Okay, so Darwin Core, you go back to that, sorry. Just clicking on it, yeah, that's good. So remember Darwin Core were all the standardized terms. Basically this is all of the data that you might be interested in. So if you only want locality or you want all terms, let's go to all terms for this one. So this gives you every single thing that this institution has made available for this record. So you can see the preparation type, you can see who collected it, and then interestingly, remember where this was showing up under MVZ, and the reason is because Tamara and Natine and Julian, which many of you probably remember, he georeferenced this when he was working here at MVZ. And so it had MVZ in the remarks field. So if you're interested in searching on a collection and you only want that collection, then there's an advanced searching and we can show you how to do that. But if you want to do this full text search, it'll pick up a name of anything anywhere in any field, even in these like georeferencing remarks and locality remarks, etc. So, okay.
So if there's data in a Darwin Core field, it shows here. If there's no data in a Darwin Core field, it doesn't show up here. So it's only populated fields that show, just to clarify.
I think we were going to do an advanced search. So you can either do advanced search up the top there or right here, there's a little advanced search. And there are a lot of things available on advanced search, thanks to Aaron. So you can pick, let's say you're interested only in things that have tissues. It's a very common kind of search people want to do. We're going to look at some owls, so bubo, let's put in bubo, that have media. So we're just looking for things that either have sounds or pictures. And I think that's it for right now. So again, we get the same list of records, and you can click on one of them. And so these are from the Macaulay Library, the Cornell Lab of Ornithology, and you can see that this does have a media record, because that's what I was interested in searching on. You can click on that, and it goes to, we're not going to play this for you, but this would play an actual song that was recorded and put in the Lab Raveau for the snowy owl.
If you've got images of Arctos and it's linked to Arctos, it'll take you to the Arctos page with all the images.
So it goes back to the original source for the data. So, okay. We can also do a filter on this.
Erase. Oops, sorry. Should we do this within a spatial query?
Yeah, let's do it. Sorry. So there's a couple different kinds of sort of filters you can use with the data search you've already done or with a new search. And so as we already showed you with Ellie's presentation, you could before do these sort of box-like searches, and now you can actually do a search that consists of a circle. So if you hit within the space of search, it'll show up at the map. It gives you a circle that you can search on, and you can change the size of that to include as many points as you want that you're interested in. Let's say you're just interested in Central California. So, one thing I didn't actually mention earlier is the map. When you're viewing the map, you will only see the records that you've just searched on. So let's say your table had 20 views in it. You're only going to see those 20 records at any one time. To get more, there's actually a load more button, which we'll show you in a second. So, okay. Is there something else we were showing here?
Yeah. Well, we were going to try to embarrass Ted, but I think given time.
Okay.
One thing that we would like to just let you know is that in a future version, perhaps in the next version, we'll be releasing a means for you to submit issues directly via the portal if a record is incorrect or needs some kind of notice. And so right now, just to give you a sense of what that's going to look like, this is not quite ready. If you search for something, MVZ is the only one that's set up. When you go to that record, you'll see a new thing up here, which is a flag. And all you'll need to do then is click that if this record needs to be updated in some way. You will, of course, you're always able to go and contact Rory directly, for example, and just send him an email. or send Carla an email directly. But if you'd like, you can log in. If you're a user of GitHub or would like to be a user of GitHub, which is a collaborative code repository on the web that we use quite a lot, you just need to then click this to log in or to create a new account. Once you've done that, you'll see kind of a scary looking page that looks like this, and all you really need to do is go down to the bottom and select Allow Access. And, oh, I've forgotten my password. At any rate, once you've done that, you'll have the ability to look at, say, the MVZ repository and all the various pieces of that, and you can subscribe. You'll also be able to see the issue that you've just submitted in the repository, and you'll be automatically subscribed to that issue, so that when Roy or Carla or somebody then responds to it, you'll automatically get notification that either actually what you're saying is right and we fixed the record, or actually it's not correct, this is what you're saying isn't correct and here's why. And so it allows for some conversation, and as well as archiving that conversation, so we have a record of those sorts of things. And or they may just ignore you, but I guess that sort of depends on whether you've got your dissertation done or not.
Dave, just a quick question. How many people here are familiar with GitHub or use it maybe on a day-to-day basis? So, okay, fair number of people.
Yeah. So if you've got a login, all you need to do is log in under your current piece, and once you've done that, or once you sign up for a new GitHub account, it's basically seamless. It'll be just like having a VertNet account that you can use GitHub to create issues and send folks information.
And it will send issues the first time to those collection managers and people listed as the contact. So they don't necessarily have to have a GitHub account already. They'll get a notice that there was an issue with this record, here's the problem, and then they'll keep getting those kind of emails as updates.
I think the key take home is you'll be able to essentially submit issues right from the VertNet portal and never go to GitHub. GitHub will just be working in the background.
Okay. So that's a little teaser for what the VertNet portal is, but please go and look at it, portal.vertnet.org.
This concludes our tag and our team, so if
Yeah.
there are any questions, we can take those.
I mean, are you using the collections designation or are you standardizing it?
So in terms of higher taxonomy, we can make some corrections and suggestions, but in terms of the non-higher stuff, we're pretty much working with what is given to us, because that's a quagmire that we're not yet equipped to wander into right now.
We're
So-
fixing, like, if there's a spelling error where they obviously spelled the genus or species wrong, those kinds of things are being fixed with
But
the
like if
migrator.
the genus changes, right? Some
Yeah, we're
collections
not-
have
Right.
60-year-old taxonomy and some of them are more
Yeah.
up to
We're
date.
trying to be-
Unless you know, you'll never find all the records.
Right, so we're trying to be sensitive to the source and allow people to control their data as is appropriate. And so if there's something like a genus or a specific epithet or something that's changed, we might be able to notify the source, but the source really needs to be the one to change it. And so that's up to them. Yeah. We know that you would be all over that here because you're like that, but who knows about the
For a lot of
policy.
reasons, practical and
But that's a big issue for sure, and one that we're not poised to address directly.
But there are standards that list current genera, current names and synonyms that you can at least use as a thesaurus to inform people. You may want to look under all of these other names when you're querying for a particular text.
And that's one of the things that Victoria and Gaurav did, is that tool they use, IDIS, and they use GBIF, and it's an excellent tool for that. And we do look at those resources for use in the migrator as well.
Well, I'm not thinking about you guys so much, I'm thinking about users
Yeah,
of
for sure.
VertNet, informing them, because most people who are users today are not either aware of or not concerned about synonyms
Right.
that are perfectly valid names that may be in use in a bunch of collections.
Yeah, no
And
doubt.
that's the value of these sources, like mammal species of the world, for example, that lists all of those.
I mean, like what would be great is if you know, like Avibase, for example, has all the bird synonyms in it.
Yeah.
So if you had a way that if somebody had a query and
Exactly, that's
somehow
what-
linked to Avibase and said,
That's what I was.
these are 17 other
Yeah.
names you may want to search
That's
under.
what I
Yeah.
was.
That's
Right,
something,
you know.
We've talked about that as a possibility, and we'll probably revisit that. It's a matter of what
There
we
are
can
some
accomplish for time. Yeah.
resources.
Do
Yeah, absolutely.
online versions exist for other vertebrate groups?
I know birds.
I don't-
but,
Yeah.
Well, mammal species in the world is online.
Yeah,
It is.
Yeah.
Significant species of the world.
Yeah.
and,
There's
yeah,
still a
the
lot of
reptile
discrepancies
database.
between all of these resources too. I mean, there isn't any, like, one standard for vertebrates, so,
But
It's a
no,
tricky-
but taxonomy is the Achilles heel of the
Yes.
whole thing.
Yeah.
Yes.
On the Chytrid issue, is your hypothesis about southern Mexico, is it compatible with this thing moving around like to Australia, for example?
So I didn't get a chance to look into Australia exactly. I know that occurred after the southern Mexico thing. So I think I've read about how they've sort of pinpointed the Australian spread to like sort of one central point and then spreading outward. And so it does.
As long as it's after your time frame, it's compatible in the sense you can use the same techniques.
Yes, yeah, so it's definitely compatible, and it happened after all of those events. I think it was in the 80s.
David, how long is this going to be in the beta version?
No longer than 7 months, because then our funding ends. But it may come out before that yet. We just have to see where we are. We just want to make sure that the foundational code is done before we do that.
Are you getting the feedback from people?
Yeah, we're getting good feedback from folks,
So-
especially people who like searching for things in vodka.
Who maintains this into the future? Beyond the
That's-
7 months?
That's a great question, and I feel like I should have paid you in advance to ask that question. We have submitted a grant to NSF for a sustaining grant that would allow us another 3 years to both develop a formal plan to distribute VertNet into the community, The way that the digitization and informatics within the biodiversity realms and in our community are changing, the rate at which they're changing is just amazing. And NSF has put a lot of eggs in their basket with ADBC and Big Bio at Florida and Florida State. And I think things are sort of headed there to be centralized, but there's also the Networked Integrated Bio Collections Alliance, which is NEBA, which is something that the community has been talking about for a long time, that could in the next 4 or 5 years become a real thing. But ultimately what we're looking to do is first get this grant so that we can very gracefully sort of move things out, distribute it, and pay for this over the next 3 years. But then also be very verbose about the fact that while we love making all of this data available for free, the fact of the matter is that it's not free. And then it costs money for Laura Russell at KU and for Aaron here and for me and for other people to maintain and to build and to support these sorts of things. And that the community in some way is going to have to support those costs, even at a minimal level. And how that will work out, we're not sure yet, but that's going to require a much larger conversation in the community. In the meantime, we've built this in such a way that it can run at a minimum sort of life support level pretty well for quite a long time with some minimum input. We've reduced the cost of the networks by an order of magnitude.
It's
We're
like
talking
$30,000 a
about-
year or something.
Yeah, we've
is the
gone
total
down from
cost.
about $200,000 annually to about $30,000. So that's a big step, but it's still $30,000 that needs to get paid somehow.
Thank you very much.
Thank you.